This is a poster at NeurIPS this year, and as the title suggests, I'm going to be talking about domain generalization and particularly for cancer classification. Right. So the problem we are working with is single domain generalization, which is a bit more restricted setting than domain generalization. So what you see here is the data that I'm working with. It's called Chameleon 17. It is basically breast cancer metastases in the lymph node sections. So the cancer was in breast tissue, but then it metastasized, so it moved to a lymph node right next to them. And what you see here is the data from different hospitals. So this data set I picked because it was one of the very few ones that have like different domains. So what you see here is one tile. So each data set, like each hospital has a bunch of slides, and then we tile the slides into smaller tiles that we work with. You see here is one tile from each center. And as you can see, they all look different from each other. And we are working with single domain generalization, which means that I want to train a model only on one of these hospitals and then have it generalized to all the other hospitals, right? Which is also a bit more practical because hospitals, like the data from, even if you're working with the same cancer type, like the data varies a lot from hospital to hospital, and of course also from country to country because of the way people stain the tissue. They create slides, how longer you wait to stain, and many other things, and scanner you use. So in our paper, we are considering a hospital to be a domain, which is like a very restrictive setting. Okay, so what this means is that if, for example, if I'm training on this center, so if I'm taking on training my model on data from this center, then this will be my train set, and then rest of the centers will be my test set, right? The validation set comes from the train center itself, of course, right? Okay, so before moving on to like our idea, I'll talk about like how people commonly go around domain generalization in histopathology particularly. So there are two common solutions that are popular in histopathology. One is simple data augmentation, so particularly photometric data augmentation. So if you have an image and then you augment it with something like this, so it might look something like this. It will look different in every single epoch, of course, based on the randomness. And the idea here is that if you change the colors and like the other information, you can also blur the image a little bit. The idea is that models should become agnostic to these things and maybe learn the features that are actually important to detect cancer. Another popular approach is something called machine code normalization. This is very specific to histopathology. For data augmentation, of course, you can use in like with any kinds of images. Machine code normalization is very specific to histopathology. So what they do is that you take, for example, I have 3 images from 3 different centers. You normalize the stains. So the way this image looks is that, or the way staining works is that you have this image, you have a tissue. And the way you stain is that you use two stains called hematoxylin and eosin. So hematoxylin stains the cell nuclei with a purplish blue color. So this nuclei, the dark spots that you see are nuclei in the cells. And eosin stains the extracellular matrix and cytoplasm as pink. Right. So outside of the matrix should look pink. And of course, they're going to be a mixture of these stains for like stuff that doesn't fall in either of the category. Okay, so what machine code normalization tries to do is that it tries to extract these two stain vectors like mathematically from a tile, from a given tile. So it's going to be this target tile. So you pick one target tile. And then you sort of try to project these colors back onto the same stain. So you basically change the color of all the rest so that they all look same. And of course, you need to do this also during test time, not only during training time, unlike this approach where you augment the data only during training time. So if you do machine code normalization on these tiles, for example, using this as a target tile, you get these images, which all look very similar, right? So you have taken care of sort of color information. What is common among both these approaches is that they both modify color information. So they don't do anything else. They just modify the color either during training or both during training and testing. Our idea is a bit different. So what we want to do is that we want to learn domain invariant features. So domain invariant features meaning that we want to learn features that do not change across domains, right? So instead of like trying the model to like sort of trying to get the model to like ignore the features that you think are not important, like just rightly get the model to focus on things that is important or that we think is important. For example, it could be the shape of nuclear structures or something else. And then the question is like, is the model already not focusing on shape? And the answer comes from like a little bit from the literature. So there was this paper from I think 2017 or 2018, a really popular one, which was ImageNet trains on CNNs are biased towards texture, and increasing shape bias improves accuracy. And what they showed was that, as the title says, like the CNNs are biased towards texture. And they showed it in a very interesting way. So the way they showed it was that if you take an image of a cat and you try to classify it, neural network will say it's a cat, as you would expect, with a high probability. If you take an image now of an elephant, but you only take the texture of the elephant, not the whole elephant, the network can still classify it. So you can still say this is an elephant with a very high probability. Now what they do, now this shows that the network can recognize texture, but does not show that the network is biased towards texture only. So what they did was that something very interesting. So they use file transfer. I'm assuming many of you know what file transfer is. So they transfer this texture from this image to the cat image, and you get something like this, and then it still classifies it as an elephant. Whereas for humans, like the humans, when they tested humans classify this still as a cat. Right? So this showed that the networks are biased more towards texture and less towards the shape. Okay, then our idea is like, why not focus on the shape that remains the same across the hospitals? So because if you look at here, like they look at different colors, but if you look at the shape of the cells and nuclei, like they remain same. Because the tissue is a 3D structure, so no matter which angle you take the photo from, like it's going to look more or less the same. Right? Instead of like natural images, for example, if you take an image of a cat from the top angle, it's going to look very different from if you take the image of a cat from like a side angle or something. Right, so we wanna focus on the shape. Then the question is like the shape of what? And in this paper, we are saying shape of nuclei, which leads to the hypothesis in the paper, which is that nuclear morphology and organization are sufficient to detect cancer. So what we are saying, this is not very clear because what we are saying, so we know from biology that when we get cancer, that nuclear morphology, so the shape of nuclei and the way they are organized in the cell, it changes. But we don't know if it has sufficient information to actually detect cancer, because when humans do it, so when pathologists look at a slide and they try to detect cancer, they look at more than just nuclear morphology. So they look at like the whole tissue, all the cells, and like the everything they can look at. Right, but we have some evidence that it can work because there's something called Nottingham grade, which is like a grade used to grade breast cancer, and then part of Nottingham grade includes looking at nuclei. So it's not the whole thing, but it includes looking at nuclei. Okay, then the question is how to get CNS to focus on nuclei. Right, so I want to make something more clear here. So one is that, of course, we are going to use nuclear segmentation mask. So nuclear segmentation mask means that we have some kind of binary mask which shows sort of like white region for nuclei and black for everything else. What we want to do is that we want to use a segmentation mask only during training, which means that segmentation should not be required during inference. So the idea here being that you should not require any extra components using our method during inference time. So it should. So if you train a ResNet 50, like ResNet 50 should be able to take, should be able to work just like a regular ResNet 50. It should take image as input and output like cancer or no cancer. It should not require like anything expensive to run during inference time. Right. So no additional components and no nuclear segmentation models or anything. Okay, then coming to our algorithm. So the way it works is something like this. So this is the regular case, the way you would do it. So you would have a ResNet 50, and I'm putting the gaps, so global average pooling layer and classification layer outside here, because I'm going to be using the embeddings here. So this is the ResNet 50 without the global average pooling and classification layer. If you take then an image and train it with the BCE loss, then this is what you would regularly do. So this is the ERM approach, normal approach. What we do is that we also take neural segmentation masks, and then we also train on another BCE loss, and then you add these two. So this is good, but this is not sufficient. If you train it on nuclear mask, so the network now like sees, okay, this is a natural image, and it can also like make predictions based on this binary image. But then what we do, and this is the main thing in the paper, is that we also take the embeddings of these two images, and then we reduce the L2 distance between them. So this now works, but the problem here is that it's not very stable. I mean, it's not a problem, like it does work, but to stabilize it a bit, so what two things we do is that we add this nuclear distance to the loss only after 5 epochs. So for first 5 epochs, we let it warm up with natural images and these binary images. Then we add the distance, and after we add the distance, we let it stabilize for 5 epochs, which works, but it's not like it was not that stable. So what we added was one thing additional, which is that we augment this image. So instead of showing the network this image all the time, because if you just look at these images, like as a human, you can see that this is a binary segmentation mask because we know it. But for a network, these two images look very, very different, because this is just a binary image, black and white, and these are like colors that you don't even see here. So it's very hard for a network to figure out like what are we trying to do with this nuclear distance here. So to make it easier for the network to reduce the distance between a segmentation mask and something that it can understand, we multiply the segmentation mask with this image, and then we show it to the network half of the time instead of the original image. So 50% probability, the network sees this image, and 50% probability then sees this image. Now it's much easier for the network to reduce the distance between this image and this image rather than this image and this image directly. So this helps us stabilize the network. And this is the whole method. So it's very simple, it's fairly straightforward, and I'll show you that it works pretty well. Okay, so coming back to the data set that we work with. So I'm going to show you those on Chameleon and then two of the data sets that we work with. So if you take the basic center, if you take the basic method ERM, so in the literature, it's common to refer to the baseline as ERM, as risk minimization. So this basically means that you just train a resin 50 without anything additional. So just like a regular classifier on the data set. What I'm showing here is that if you train an image, if you train a model on images from center 0 and you test it on all the other centers, this is the accuracy of that center. And we train 10 models per method per center. So there are 50 models in here, one for each center. And then you average the accuracy. So you get the average. So 62% is okay. And here, by the way, I'm not using any augmentation. So the only augmentation I'm using in the methods that do not say -o-aug aug is random horizontal and vertical flips. So just geometric augmentations, but no photometric augmentations here. Right? If you do use Mashenko, so this is a normalization method I showed in the beginning, which sort of changes all the images to like the same color. It works pretty well. So it improves the accuracy quite a bit. So it goes from 62 to 73. And then you take a bunch of other methods. So for example, one method that one of the reviewers asked us to do was to take HooverNet. So HooverNet is the network that we use to generate nuclear segmentation marks. The idea was that if the network can segment nuclei already, so the encoder should have knowledge about like where the nuclei are. So maybe then this is sufficient and you don't need your algorithm to train it. So we just use the weights from HooverNet as initialization. This works more or less same as ERM. And then these two methods are from outside histopathology. So they were developed for natural images, but they seem to work pretty okay, even for, especially L2T works pretty well actually, much better than Mashenko even for histopathology. And then if you take our method, it works really, really well. So it's like 12% better than almost 12% better than the next best method. And this is without augmentation. So when you add photometric augmentation, then the things come closer. So ERM like accuracy all jumps to 85, and then our method like also see some increase in accuracy, but it's not too much because it's probably the accuracy is already kind of saturated. But the difference is quite a bit. And then we have two ablation studies here. I'll show more ablation studies later. One is that if we do not use L2 loss, so we do not use L2 loss, but we use the augmentation of the one I talked about, which 50% image versus 50% nuclear mass times image. So then you have like something that is better, but it's not like you still need L2 localization to actually learn the nuclear, to get the network to focus on nuclei. And this is without anything. So this is basically ERM, but you also train basically ERM plus the additional BCE loss on the binary mask image. So without L2 loss and without augmentation, right? Okay, so besides testing on these, since you can always overfit on the training set, so it's becoming more and more common in histopathology, at least, to test on external test sets. So what external test set mean is that we use a test set which we do not see during our project at all. So I tested all the models from all the methods exactly once during the whole project, so never again. To sort of show that, okay, our method and our models are not overfitting on this data set. So what we do here is that I could not find any data set that had this exact same setup. So exactly like the same lymph node sections that has tumor from breast cancer. But I could find like other cancer data sets, and it sort of seems to work. So this one, for example, this one is BCSS is the first one. This is primary breast cancer. So the image, so again, it's breast cancer like our data set, but it's not lymph node section. It's now in breast cancer itself. So the tissue looks like the breast cancer tissue. Not like the lymph node tissue. And this is from TCGA, and it has a lot of tiles. So we are testing on 108,000 tiles. So for those of you who are familiar with histopathology, we are not reporting site level accuracy, we are reporting tile level accuracy. So quite a bit of tiles to test on. And then if you see the accuracy here, like we see the same effect. So accuracy goes down from 91 to 78, which is better because the tissue is completely different. But you see the difference here still. So our method is still like without augmentation is still like pretty good, like pretty good and like pretty good for a model that would generalize from lymph node to breast cancer, close to the second best method. Another test we use is Oslo. So this one is even more differentiated, like even more different from the first one. This one has cancer slides, but from completely different organs. So nothing to do with breast. So there's bladder endometrium and all the other organs. And this has many more tiles, so 174,000 tiles. And what we see here, what we saw here was that there was a difference. So you still see the same trend in general, like the accuracy of our method is much higher than the close to the second best method, but difference here was not too big. And when we checked the numbers, we saw that the difference was, this was because of one organ here, which was kidney. So kidney here is, one thing is that it looks very different. It's clear cell carcinoma, which looks very different from like other carcinomas, but also that it had the most amount of tiles. So it was like, it amounts for most of the accuracy in this, not most, sorry, like a big percentage of accuracy in this case. And the highest accuracy on kidney was, for any method, was 64%, while for other organs, it was at least 70%. So kidney was basically bringing down the accuracy for all the methods. And then if we check accuracy without kidney, then you see the same differences again. So it's like our method is again like about 2% better, and these, like, all the accuracies jump up a little bit, right? So sort of these data sets sort of show that our method works, so we are not overfitting it on chameleon data set, basically. Okay, I'll now move on to ablation studies. So ablation studies, I'm abusing the term a little bit here. So it's not just ablations, like analysis sort of. But what we're trying to show here is that our method actually is focusing on nuclei. So it's not like some kind of regularization that is helping, but it's actually focusing on nuclei itself. And we are showing that in many different ways. So first one is by subtracting the embeddings. So what we do here is the opposite of what we do in training. So instead of reducing the L2 distance between these two, you take the embedding of this image, of the original image that you want to test on, and its nuclear segmentation mask, and then you subtract the embeddings. So the idea here is that you have sort of removed information from this image about the nuclei, if the network really is learning like the features that are very similar to this this image, which is what L2 distance is trying to do. And then you check the accuracy. So what we expect is for our accuracy for our method to go down a lot, which it does. So it goes down to below 50%. So 50% is, by the way, is what a naive classifier would classify, because in our data set, we have exactly 50% tiles that are tumor, 50% that are not tumor. So if you take classifier that classifies everything as only tumor or non-tumor, you will get 50% accuracy. So our method is really bad. Other methods are like, since you subtract something random image, so other methods have never seen this image during training, right? So for them, subtracting some kind of like black and white image is sort of like subtracting random features. So their accuracy decreases a little bit because you're disturbing the statistics of the methods. But it doesn't go, it's still like quite a bit above chance. So they are still performing fairly okay. Next, we tested on like these images. I'll get on them one by one. But the idea here again being like, we are trying to figure out whether it's focusing on nuclei or not in many different ways. So first way is that we remove all the information from the image, except we keep the background. So we keep the background in the nuclei. So you multiply the image with the nuclei, but you make the background, instead of black, you make it white, because white is also, if you look at these image, for example, the background in the default background in the tile is white. So basically the methods just see nuclei. So if the method can infer something from, if the model can infer something from just nuclei, then it should perform very well. So which is what we see for our method, like it performs almost as good as like with the original image. For other methods, they go down, the accuracy goes down quite a bit. So because it is relying on information also from other than nuclei, right? Next thing we do is that we remove, so one thing could be that maybe our method is looking inside the nuclei as well. So we don't want it to look at the texture at all. We want it to look at just on the shape and the organization. So we remove this intracellular, internuclear information by just coloring the nuclei. So if you color it black like this, now it's just nuclei on the white mask. And then, of course, our method is still performing fairly okay. So which shows that it's not relying on the information inside the nuclei. So it's relying really on the shape, size, and organization and stuff. Other methods are, of course, not performing too well because for them, this is a very weird image to look at. Same just for completion, like same with the mask that we train on. Again, our method actually performs slightly better than without this image. So then on natural images. Other methods like almost fall to random chance because this is a very weird image for them to look at because nuclei are never white in the images that they have seen. Then we do the same thing. So we remove the intranuclear information, but then we introduce this background. So methods now can look here also. So the background is like more, you have the tissue as well to look at. And accuracy for our method is again about the same. So it's always hovering around 91. For our methods, like they are not doing much better now because they have information that they can actually look at, right? Other than nuclei. So they are performing like not as good as they were performing with like the nuclear, with the original nuclear, original nuclei in their original color, shape, and texture, but like it's still fairly okay. So for example, L2D, the next best method drops from like 89 to 84, but not beyond that. Okay, then we make the nuclei white. So this is interesting because white is also the color of the background. And this is also nuclei. So our method now sees this nuclei as white nuclei, but it sort of can confuse the nuclei for the background, which we see that it does. So the accuracy is there because you can, as a human, you can still see like there is some boundary around the nuclei, but it's not entirely clear if it's nuclei for a model. So the accuracy is better than other methods, but the drop is not as high as for other methods, but it still drops quite a bit, which is showing us that it does need to look at the nuclei. And this is one of my favorite ablation studies in the paper. So here we are like basically doing what we would want to do ideally to see if the network actually rang on nuclei or not, which is that we in-paint the nuclei. So we in-painted using OpenCV, simple. And then here you have completely removed the nuclei. So the idea here is that if the method is relying on anything at all other than nuclei, then the accuracy should be at least something. Right, and what we see here is that here I'm reporting only in-domain accuracy, like we have all the accuracies in the papers, of course. Out-of-domain is like even worse. But in-domain accuracy, so accuracy on the validation test set is like very high for, not very high, sorry, very okay, so above random chance for other methods. So if you look at the method string documentation here, it's like above 60% for all of them. For ours, it looks like it's 48, which is below random chance, but not too bad. But when we looked at the predictions, we saw that our network was basically predicting everything with a single class. I think it was predicting everything as non-tumer, which you can also see when you look at recall. So if you look at recall, for our method, it's basically 0. Right, so it's not able to predict anything. It's just saying every image is basically looks the same without the nuclei, which is again goes on to show that our method is indeed looking at nuclei, not anything else. Okay. Even after all of this, like the reviewers, when the expert reviewer in the nearest was still somehow not convinced that, or like was convinced but not fully convinced that our method is indeed looking at nuclei. So the reviewer asked us to also generate saliency maps, which we did then. So I have saliency maps, and I'm using integrated gradients to show sort of where the network is looking at. And if you look for a baseline method, so ERM, then you see for this image, like this is the contribution from the positive class. This is the contribution towards the negative class. So green is positive, red is negative. But it's looking everywhere, and this is what you would expect. And I'm not cherry picking the image here. Like all the images I looked at, like I looked at a few hundred images or something. They all looked basically more or less like this. So for our method, it looks only at nuclei. So if you look at the boundaries here, like it's focusing it's looking exactly at the boundary of the nuclei, right? Both for the positive class and the negative class. So our method is indeed it drives the point home that it's focused only on nuclei. Okay, this was ablations. So now I'll move on to robustness. So beyond showing that our method actually increases accuracy on the task at hand, we also show that it increases robustness in 2 different ways. So first one I'm going to talk about is image corruptions, and then I'm going to move on to adversarial examples. So image corruptions we consider, so there was a paper that showed this had a bunch of image corruptions that people test on natural images. So we took the same ones, and then it's a bunch of these. So for example, if you compress the image from PNG to JPEG, so you lose some information, blurs, noises, and also snow. And also, by the way, lattice transform, which sort of like changes the shape of nuclei here, as you can see. So like it changes the shape of everything. And the most relevant ones for in practice would be defocus blur and motion, because sometimes the scanners do blur the image a little bit. So this is like practically also like more relevant, I guess. But they're all sort of show whether the models are robust or not. Okay, then if you compare our method to like the second best performing method, L2D and ERM, which is the baseline, our method on an average is better. So L2D performs better on some things. For example, particularly like these 2 noises, it's slightly better here and here. But other than that, like our method, the difference is like, especially if you look at the blurs and elastic transform even, our method is better. This was image corruptions. Then moving on to adversarial attacks. So adversarial attacks, for those of you who I think everyone knows, but if in case you don't, like the best way to explain is with this meme. So the meme is saying basically the corporate needs you to find the difference between this picture and this picture, and Pam, the human, is saying they are the same picture, which is what would happen in real life. Like if you look at this image and this image, as a human, you would say that they are the same image, but actually they're not the same image. So one of them has been adversarially generated to attack the model. So these are the 2 images, and this is I don't know how clear do you see it on your screen, but this is the difference between these two images. So if I subtract this image from this image, this is the difference that you see here. It's very slight, it's very random, it's very slight, it's very small, but it's enough to fool the network into thinking that this has tumor versus this has no tumor, right? So the reason we thought that our model might be more robust for adversarial attacks was that because since it is looking at very few things, it's only looking at, like, as you saw from integrated gradients, like it's looking only around the nuclei and the organization. So there's like less surface for the models to attack the image, if that makes sense. So you can attack the method only by changing the nuclei more or less, or that's the hypothesis that we have. If you change the image elsewhere, it might be more difficult, right? So if we look at and we are using PGD to attack them to attack the models. So if you look for our method is at the top. We are using 6 steps, which means that we do the backpropagation step 6 times, and then the alpha is the step size is 3 out of 255. So 3 bytes. So people choose 255 because 255 is like the number of pixels. Sorry, the pixel range that you have. So this is the typical way to pick alpha and also EPS. This is the parameter specific to PGD. Okay, for our method, like you can clearly see that it's more robust. Of course, if you increase the EPS too much, accuracy, all the accuracies do go down to like 10%. This is one way to look at adversarial attacks, but the more interesting for me was to look at cross-model attacks. So in cross-model attacks, what we're trying to do is that we try to generate image using model trained on one method, and then test those images on other methods. So in this way, sort of, we are comparing methods head to head against each other. So we can see if one method can attack another method in some sense. To explain it more clearly, so for example, if you have this image and you have a model, let's say, so this is the baseline model, ERM, what you would do is that you would take the image and then you pass the image, you get some label, and then you flip the label. And then you propagate the gradient backwards, and then this would generate an adversarial image. And you would repeat, this image would act as the new image, and you would repeat this process a couple of times. So steps, so we are doing it 6 times. But what you're doing if you took this image and you test it on this model, this was the previous graph. So then you're testing it on, basically, you're seeing if the model can attack itself. But what we are doing here is that we are taking this image now, this adversarial generated image, and we move it down, and then we test it on, for example, our method. So now you would pass the image to our method and then check the accuracy for our model. This is what we're trying to do in this experiment. And the way I'm going to show it is, so since there are too many methods, we are testing it again only for 3 methods, which is the ERM, the baseline, then the second best performing method, which was L2D, which was way better than all the other methods, and then our method, of course. So if you look at the lines here, the first line, the line at the top, is the accuracy of ERM if you test it with images generated by our model, and then the dotted line is one if you test it with L2D. So as you can see, L2D is able to attack it slightly better, probably because it's not trying to change more than just nuclei. But they are both able to attack the model. I have no idea what is happening here. Why is this accuracy going up suddenly here? But I saw this across for all the methods, as I will show. Okay, if you take L2D, its accuracy is of course better. It's more robust than ERM, but our method is still able to attack it, and the accuracy still goes quite a bit. And again, there's this bump here. But if I take our method, like, since again the idea here was that since our method is looking only at nuclei, maybe it's harder for the methods to attack it. And this is what we see here. If you look at our method, like, accuracy doesn't go down much at all, at least from ERM. For L2D, it goes down a little bit, but not that much. So our method pretty much stays at the very top, right? Again, showing that our method is more robust. And yeah, that's it. Thanks for listening. I'll now take questions.